So, welcome everyone to the afternoon session of this symposium on how microbes rule the world. We have another number of exciting presentations in front of us. I'm sure of that. I'm Lars Tranvik, a member of the Crafoord Prize Committee, and I will be the chairperson for this afternoon. And I will just start by inviting the first speaker, Ramunas Stepanauskas, who actually got his PhD here in Lund in, I think, year 2000. And he is now at the Bigelow Laboratory of Ocean Sciences in Maine, United States. Welcome, Ramunas. You will talk on 1 drop of seawater, 1 million organisms.
Thank you very much, Lars. Thank you, Crafoord Foundation, and tack så mycket för inbjudan. And congratulations once more, Penny. It's this award is definitely belongs to you 110%. I work at Bigelow Lab for Ocean Sciences, which is situated in Maine, northeastern United States. Actually, that area looks very much like Sweden. It's beautiful. You should consider coming for a visit. And every summer we have several events for the general public, including open house and some Café Scientifique events. And I recall a few years ago we were chit-chatting one of the visitors who just stumbled into that event by chance. He clearly didn't have anything better to do, so he stopped by and was totally perplexed by these grown-ups with their PhDs spending their lifetime studying marine microbes. It just seems like, now if you have the smarts to be a scientist, why don't you do something important? And he wasn't mean, he was just totally puzzled. And you can't, I think, blame him, because that's probably what most of the people think, and I don't recall myself growing up dreaming to become a microbial oceanographer. I don't know, Penny, did you think of it in middle school? No. Yeah. Anybody in the audience? No. Okay. Yeah, they're just so small, who cares? And counterintuitively, we have to think really big in order to appreciate these tiny critters in the ocean, like having a look at the planet from some distance. And essentially what it is a huge ball of molten rock that has crusted up at the surface, and most of that surface is covered by the ocean. That ocean is full of life, and most of the life there is microscopic. It's unicellular, bacteria, archaea, and protists. And these microbes have been doing all kinds of things to the planet for almost 4 billion years, like producing oxygen that we breathe, organic material that the rest of the marine biota rely on as their food source, and we humans take that livable planet for granted without even noticing the workers behind the scenes. Now, if you are a marine microbe, you would probably think that there's hardly anything else on the planet besides other marine microbes, and that point of view is probably more balanced than our point of view. The main reason why we don't fully appreciate this, the significance and the complexity of microbial life in the ocean is, to a large degree, the limitations of our research tools. Penny's discovery of prokaryoplankton is actually a good example of how important is technical advance to the advancement of knowledge in our field. Now we know that prokaryoplankton is the most abundant photosynthesizer in the world. 3 decades ago, we had no idea that it even existed. And that's because Penny started using a new type of instrumentation called flow cytometry to analyze marine samples. Today, some of the most novel insights into marine microbiology comes from various molecular tools which we collectively call Omics. And that brings us back to year 2000, which was really an extraordinary year for biology, because for the first time, we sequenced and assembled almost complete, an animal's complete human genome. That took more than a decade of work by thousands of scientists. It cost $4 billion. It was a huge project. But when completed, we had this reference genome of a human that allowed all kinds of other molecular tools to be developed and serve various purposes, like comparative genomics, transcriptomics, proteomics, and others. And that's because when you have a reference genome, you can start mapping relatively cheap, easy to produce short reads of DNA, RNA, and proteins to that reference. And today, these various omics tools are really important in various areas of not just research, but also in practical applications like diagnosing some diseases and also getting into treatment of them. So our field, microbiology of oceans and other environments, was quick to embrace these new powerful tools and start applying them in all kinds of complex microbial communities in bulk. And we started calling these tools metagenomics, metatranscriptomics, and metaproteomics. And today, they're also a super important source of information about the function and especially about the composition of various microbiomes. Unfortunately, there's a big difference between the omics of humans and the meta-omics of microbiomes. And that difference is that we never built that foundation of reference genomes. And that puts some serious limitations to what we can actually do with the meta-omics data from microbiomes. So one example here is a recent global large study of oceans called Tara Oceans discovered that surface ocean microbes alone encode about 40 million gene types or gene clusters, which is really mind-boggling. That's 2,000 times more gene types than the number of genes in the human genome. There's no question that many of these genes are important for the planet. There's no question that many of these genes could come handy treating diseases, coming up with new bioenergy production solutions, and other practical needs for humans. Unfortunately, only 0.5% of these gene clusters can be mapped on references, which means that the vast majority of this data is very hard to interpret. We don't really know what these genes encode. We don't even know what kind of microbes they come from. It's kind of like going to Las Vegas, winning $40 million, and then losing 39.8 of these millions the same night. So 2.5 years ago, I was lucky to get support from the Science Foundation to start working on a fairly large project that we would try to address this big problem. And our approach here is large-scale randomized single-cell genomics. We started with the samples from the tropical and subtropical regions of the ocean in Atlantic and Pacific. We started from collaboration with Penny. And we put a special emphasis on one of these samples that comes from the middle of the Sargasso Sea, the BATS station, which is one of the 2 stations that are being hot in Hawaii. That has received tons and tons of attention in various research projects, including the very first meta-omic studies of marine microbes. And in this case, we started with this single cryovial full of seawater. It's 1 milliliter, actually. Have a sample of what it looks like. It's a vial of this size. So we started with this vial. We used half of it to produce data, and we put the rest in the freezer. It's still there. And this 1 mil was sufficient to generate over 6,000 of partial genomes of marine bacteria and archaea. They represent 11 phyla, including some of the so-called candidate divisions, or marine microbial dark matter. And since we captured DNA from all the sequences from all the DNA molecules in the cell when we apply single-cell genomics, we also captured hundreds of novel phage co-systems in the same projects. For perspective, the scale of this sequencing is comparable to the number of genomes that we sequenced by the entire world in year 2013. For different perspective, the number of genetic letters that we read from these 400 microliters of seawater is equivalent to 10,000 average-sized books. So that's all cool, but on the other hand, we only read 1% of all the genetic information that was actually present in that 0.5 mil. And that's a very useful perspective to have in mind. It's humbling, and it tells how much we still have to improve our technologies, no matter how fancy they are. Of course, there are many milliliters in the ocean. So, but yet, that's all big words, but at the end, getting this data from 1 mil was not easy, and it wasn't cheap, so was it worth it? Or were we just reading the same genetic letters over and over, which would be embarrassing to report back to the funding agencies. And luckily to us, that's not the case. So I don't know what you think about this figure. I'm going to explain to it, but it's the most exciting figure that we produced, I think. Doesn't look very much. What it shows is, so the metric here on the y-axis is the pairwise average nucleotide identities of the genomes that we sequenced. And that's a commonly used metric to measure the evolutionary distance between two genomes. And for perspective here, all the humans on planet share about 99.9% ANI. Chimps and humans share about 96%. Humans and cats, 90%. Humans and cows, 80%. So essentially what this figure shows is that for an average microbe in this drop of seawater, the vast majority of the neighbors are less related to that microbe than we are to cows. I think that's pretty good insight. Who are these microbes and what do they do in the ocean? This figure took us a lot of work, a lot of frustration, starting over and over, and it's really busy. But it's actually just a simple summary of just a few biochemical capabilities that the microbes that we recovered from these single-cell genomes are encoding in these genomes. And you're well familiar now with the prokaryoplankton and the sister groups in the coccus. They are the only groups in this data set that are capable of oxygenic photosynthesis. They also encode several bioactive compounds. Penny already talked about some lantipeptides. They actually encode even more of some less exotic compounds, terpenes and bacteriocins, which are very common small molecules produced by marine microbes. So that's for the kind of the reference point. One of the larger surprises in the study was this group of alpha proteobacteria, which we discovered only about a year ago, or described for the first time about a year ago by Ben Tully's group. It turns out that they encode all the genes that is needed for photosynthesis, but not the oxygenic one. It's an anoxygenic type of photosynthesis. And what our study shows is that they are actually really abundant in the ocean. So their physiology still has to be confirmed by experiments. Right now we just have their blueprints from genomes. But if confirmed, they may turn out to be among the most common photosynthesizers in the world, potentially only second to prokaryoplankton. We have to see still. There are other groups among these microbes in the ocean that also fix carbon, but not based on photosynthesis. They use various chemical reactions to gain energy for that process, and one of these groups is this the group with the cryptic name SAR324. In this case, they appear to be gaining energy from self-oxidation. What's also interesting about this group is that it stands out from all the other lineages by a very rich set of secondary metabolite pathways, including polyketides, which is this red color here in the pie charts. And the polyketides is kind of like the holy grail for people who are looking for novel, medically relevant small molecules. So that entire jungle, Amazon of microbial life, was pretty much hidden from human knowledge up until very recently, and it resides in just every single drop of the ocean. Now, one of the how do we judge the performance of a reference genome database, which was really one of the original goals of the project? So one of ways to measure it is by using that database as to map the existing metagenome reads on the collection of genomes. And that's what we did here, using various collections of genomes from various publications. And in this case, we used this 95% average nucleotide identity threshold, which is a commonly used delimiter of species. It's an operational definition. It's kind of arbitrary, but that's a commonly used technique. And this blue box plot here shows that the database we produced from single-cell genomics from all these 28 samples that we had to start with. On average, they recruit about 40% of metagenome reads from an average metagenome in the tropical and subtropical ocean. That's pretty cool. That's really good, considering that we are trying to represent here a biome that covers a third of the planet. It's actually 10 times better than all the marine microbial genomes back in 2013. So I'm pretty happy with that, but I'm even more excited by the fact that this single cryovial, or half of it, was sufficient to represent half of that metagenome recruitment. That's, I think, really amazing, and it shows that every little parcel of seawater encodes a major part of the entire coding potential of marine microbiome. It was also surprising to see that this single milliliter of seawater outperformed all the metagenome bins that were produced by various groups from the entire global set of metagenomes. It also outperformed a large collection of marine microbial isolates. Now, how is that possible? I think it raises some really important questions about the technologies that we are using in our field right now, and their limitations. And it brings back the question about the techniques. So there are various ways to produce microbial genomes, to read the genomes from microbes, and the general ideal outcome generally assumed would be that we want to recover all the complete genomes of all the microbial species in that sample. The traditional way to do it is to first obtain pure microbial cultures, then extract DNA from each of that culture, shotgun sequence these cultures, and then use computational tools to assemble the short reads into discrete genomes. It's like putting together a jigsaw puzzle. In this way, we can actually get really high-quality genomes. We also have the cultures to work with the culture experimentally, which adds a lot of power to the study. So it's a powerful approach. Unfortunately, most of marine microbes do not like to grow happily in a lab. And it takes a lot of work to come up with the conditions to grow yet another culture. So it takes a lot of work, and it's not scalable to the degree of diversity that's out there in nature. So at the end, what we end up with is beautiful genomes of a very tiny fraction of microbes that are out there in the environment. So one of the alternatives is metagenome assembly and binning, where we extract DNA from the entire microbial, that original environmental sample, and then perform shotgun sequencing, so we capture that entire microbial community at once, and then proceed with the advanced computational tools to assemble and bin these short reads into discrete genomes. That circumvents the need for cultivation. It's very direct, and it's actually very simple as far as lab techniques go. Unfortunately, it's not perfect, because you don't recover still all the genomes, and the genomes that you do recover usually are not complete, and they may be chimeric. And trust me, the chimera of a lion and a goat is very boring compared to some of the chimeric assemblies of microbial genomes that end up in public databases. And that's a big issue right now. So another alternative that we applied in our study is single-cell genomics. We start with a physical separation of individual cells. But then, instead of cultivation, we use various biochemical tools to produce many copies of their genome so that there's enough for sequencing. And this amplified DNA is then used for shotgun sequencing and de novo assembly of each individual genome separately. So in this way, we still circumvent the need for cultivation, and we also prevent the formation of these chimeric assemblies, which is good. Another advantage is that we can perform this single-cell sequencing in a randomized quantitative manner without any major taxonomic biases. So we get a quantitative sample of the genomes. It's not perfect, because we don't get complete genomes from every cell we sequence. We get partial genomes. Now, at least in theory, we can actually produce many genomes of close relatives. And in theory, you could take these partial genomes and co-assemble them into complete genomes and get all of the species in the sample covered, in this way achieving this original Holy Grail goal. In practice, it doesn't happen, and the reason for that is that after sequencing tens of thousands of single cells, we still did not find two that are identical. They may be related, but they're not the same, and that's a very important biological insight that we have to keep in mind, I think, in all of the studies of microbes in nature, what it means is that this original goal itself is misguided. There's no such thing as the genome of a microbial species. And we actually don't know how to define a microbial species to start with. So let's, for example, let's have a look at the size of the pangenomes, or the collective number of gene types or gene clusters in these various microbial groups or lineages that we recovered from the marine sample. Quite many of them have more genes in these pangenomes than the number of genes now in human genomes. And Penny already mentioned that. Prokaryoplankton is there. SAR11 is even more abundant, and they have even larger pangenomes. So this is an enormous genetic resource that these marine microbes have accumulated over billions of years of their evolution. And it certainly serves them in their survival. I think humans have also a lot to gain by better understanding this genomic resource and using that to the best of their benefit, both as stewards of the planet, as well as in biotechnological applications in practical uses of these genetic resources. And so talking about techniques, the data for this project was produced by the Single Cell Genomics Center at Bigelow Lab. As I mentioned, that first step in the process is separating cells one by one physically. And that separation is done using instrumentation called fluorescence-activated cell sorting. It's a flow cytometry that includes deposition of cells into, in our case, into microplates. We performed the sorting in a HEPA-filtered cleanroom to prevent DNA contamination, because that's really the tricky part of the technique. You have to be super careful, considering that each of these individual cells have only one copy, in most cases, of their genome. So to produce enough of that material for sequencing, we have to amplify DNA, and that amplification reaction, in our case, we set it using robotic liquid handling, which allows us to scale up the process. We can process thousands of cells in parallel, and it also prevents contamination from hands. It allows us to also reduce the number of errors. Human hands are more prone to errors than robots in this case. The next step in the process is taking then that amplified DNA and using as a template for Illumina library preparation. We use Illumina technology today. And then from there on, it goes into a sequencing machine. The next-gen sequencing technologies that are in use today, like Illumina, they produce tons and tons of very short DNA reads, which can be compared to a huge pile, millions of pieces of jigsaw puzzles. And so the next step that's equally important as the lab side is the computational algorithms that can take these piles of short DNA reads, which are just a few hundreds of DNA bases long, and put them together like jigsaw puzzles into the genome that was encoded inside the cell. And these genomes range in size from 1 to several millions of letters. The center itself is an open facility, so it serves not only Bigelow, but also scientists outside our institution. If you're interested, check out this website. Of course, this work was done by many people. It was a lot of work, a lot of fun too. And the credit goes to my research group, fabulous people, both in lab and in the field and in front of computer, the staff of the Single Cell Genomics Center, as well as many collaborators, and especially we are indebted to Penny Schism. The study itself actually started as an offshoot from one of Penny's own projects on prokaryoplankton. Last but not least, I want to thank our funding agencies. Without them, there would be no research to report. And Simon's Foundation is the main source of funding for this particular project. Now, giving this talk in Lund is a special opportunity for me to also thank my former mentors and teachers from Sweden. Especially, I want to thank my former mentors, Lars Lars Tranvik and Lillemor Levan, who enabled my graduate studies here in Lund. Also want to thank all my teachers at Lund, Uppsala, and also Stensunds Folk College, which we studied before Uppsala. I grew up in Lithuania back when it was still occupied by the Soviet Union, and in early 90s, when that decrepit empire finally fell apart, Sweden provided many amazing opportunities for students from Lithuania, Latvia, and Estonia, and I was one of the lucky ones. So I came to Sweden as an undergrad, and I had $20 in my pocket, and 7 years later I have a PhD in an area you love. So thanks Sweden. Tack så mycket.
Thank you, Ramunas. We have plenty of time for questions. Do I see? Yeah, Christer?
The microphone here. Can you hear me? Yeah. Sorry, a question out of ignorance maybe, but if you compare these figures on biodiversity in 1 mil of marine water, compare that to a limnic environment. This is mainly about marine biology, I realize, but there are many other biomes, habitats in the world. Could you just give some figures for comparison?
I wish I could. I don't have that kind of data. This is the first study done at this scale. We have small-scale projects of sequencing single cells of microbes from lakes, also from soils, my garden soil, but they're much smaller. We just haven't had a grant, but maybe we can write a proposal together and get a foundation buying into the idea. I think that's something that has to follow up, and hopefully also techniques get cheaper so it can be scaled up and applied to much more environments. My impression is that limnic environments would be a little bit less complex because they are younger. They haven't had these billions of years of evolution and diversification as oceans have. But there's certainly plenty of diversity in them, perhaps at a finer resolution especially. In the case of soil, the general assumption is that they're actually more diverse than oceans. I think that jury is still out there, and to figure out how exactly they compare to each other, and the trick is to figure out how do you actually measure diversity. What is your metric when you start talking about microbial diversity? Because then very quickly you have to get into questions talking about species, and then you realize, well, we don't really know what is a species, so what do we measure in that case?
Sharon?
A question regarding your reference metagenome thing. So when you say that each taxon then is comprised of a innumerable number of genomes that differ a little bit, each of them, doesn't that mean also that you don't get a reference genome, or is it so that, I mean, because if they're all different, they become useless. But is it so that some particular SAGs, or I mean, representatives account for the majority of the reads in the metagenome?
I'm not sure about just a small subset of them accounting for the majority, but actually giving that question, I do have some extra slides. And I excluded this one because it's kind of busy, but it does actually address the question of how well does it perform as a reference material to annotate a metagenome. So what we did here is we created an artificial metagenome by producing new single-cell genomes and then shredding them to mimic short reads of metagenomes, and then pass these short reads through an annotation pipeline that relies either on our database, which is this Gorge, or GenBank non-redundant database, NR. and we did functional annotation, and we can show that Gorge performs really well. You can't do direct annotation of short Illumina reads on themselves, relying on just GenBank, but you can actually with the Gorge. Over 80% get annotated correctly, and we have fewer than 5% error rate. And we can also assign taxonomy correctly down to genus level using this Gorge database. Also, more than 80% of individual reads. So it's, I think, 250 on average basis. So yeah, this is a major improvement of the cyber infrastructure for marine microbiology, because now suddenly you can take you don't have to go through the assembly first and deal with the chimeric results and huge computational need. Resources that you need for that assembly. You can just pass individual reads and you get correct answers.
Thank you. Justin?
So this is probably a very stupid question, but I mean, you have a lot of asexual reproduction here among all these microbes. So I mean, just by chance, will you not end up with the exact same genomes at least once in a while when you do sequencing drops?
Well, I mean, I'm sure there are identical genomically identical cells in the ocean. When sister cells just divide, now in many cases they may end up making no errors at all, and they would be identical. But when you actually go into the ocean and collect that sample, your chances of finding these identical cells are pretty much zero. I did a calculation a few years ago, what is the chance of finding these identical cells, and I forgot the number, but it's one of these astronomical numbers that's pretty much, it's impossible. And that it's not just point mutations, so after sequencing these 12,000 plus genomes, still with every new genome we sequence from single cell, we are still adding 45 new gene clusters with every new genome. So it's also the content of the genome, the new genes that make the difference between one and the other cell.
Yep.
I just had a question about the going from genetic diversity. You had like the cow and the human. I was just thinking about, how do you think about going from this genetic diversity in microbes to something like functional diversity if you want to try to take this to an ecosystem perspective?
How do you measure it?
I guess that's the question. How do you think about, right? So you're stuck with, you have 10 to the some large number of variables in the system, right? Yeah. So if you're trying to build a simple model of the ecosystem with, say, 5, how do you try to bridge those scale gaps as we just get more and more and more data from these genetic techniques?
Yeah, so this figure was kind of a simple attempt to capture just a few of the features, some of the important features that microbes encode in their genome, carbon fixation, and you can see that it's actually just a few of the microbial lineages that are performing carbon fixation. So if you are focusing on modeling and understanding carbon fixation in the ocean, it is doable. You can focus on these lineages. You can look at the details of their physiology and use information from genomes, from experimentation, from other techniques, and get to it. But I think what we still have to do more is the appreciation of how complex is the system and how we have to first think big and only then go small. Otherwise, we start losing the big picture. I don't know if I answered the question. It's a broad question, so do you want to follow up with some?
I mean, so you could think about viewing this as all we cared about was, I know people who work on, say, these systems as purely, imagine there's just chemistry, and we can reduce the problem to, like, say, 7 or 8 chemical reactions. So not just modeling carbon, but let's say we want to model some functional version of this microbial ecosystem, right? How many types will you actually need to do that? Can you group these into, say, 5 classes of chemistries using only the genetic information, for example?
I think you have to ask a very specific question before you start grouping them into various bins, because there are so many ways you can divide this data, and to make it really relevant to your question, you have to be much more specific in that division. So if you ask a question about carbon fixation, you have to look specifically at that. If you're asking a question about degradation of organic carbon, you would have to look at that data differently. If you look at infections by phages, which is now we know is very important, again, it's a totally different set of complexities that you have to consider. So it would be nice, to be very convenient, if you could divide the marine microbes into a small set of bins and then deal with those bins. But in reality, the number of bins is endless. Each genome of microbes has a few million genes, and you can consider each of these genes a feature. And then you go to another genome, and it has a slightly different or vastly different set of these genes, and then you have another few million of new features. So binning these enormous sets of features into just a few is not trivial, and I think it just has to be driven by some very specific questions.
Okay. We still have a couple of minutes according to schedule. If anyone has some pressing issue to raise. Yeah, Penny.
Well, this gets to the two identical.
So the microphone is right here. Oh, there's one.
They're all of them. Does it just go on automatically? This gets to the question about two identical genomes in 1 milliliter of water. Have you done the physics of, like, if a cell divides, how long it takes before it's not there in that milliliter?
Well, your group actually that calculation.
Did we do
so
that calculation?
I just trust that you did it right.
What did we say?
I think the conclusion there was that after division, within a week or two, the physical distance on average is about a kilometer between these cells, just based on kind of average speed of currents and turnover of surface ocean.
But I wonder what that means, anyway, for your little milliliter in a day. Anyway, I'll take that question home. Okay.
You can talk about it in the coffee break.
Yeah.
Thanks a lot, Ramunas.
Thanks, Lars.